Good afternoon. My name is David Wade. I'm affiliated with the Wade Research Foundation and also with New Mexico State University. The title of my talk is Names as a Source of Novel Peptides. This topic is a direct result of postdoctoral work that I did with Bruce Merrifield, the 1984 Nobel laureate in chemistry at Rockefeller University during 1988 through 1991. The concept that I will describe took about 12 years to incubate in my mind and was finalized when I heard my 4-year-old niece singing the song Name Game, which was made popular by Shirley Ellis in 1964. So by 2003, the concept had been finalized and I made the first publication about it. The logo of the American Name Society is promoting the study of onomastics. And Wikipedia defines onomastics as the study of the etymology, history, and use of proper names. Since 2003, the latter part of this definition, the use of proper names, has been expanded to develop a new branch of onomastics, one that includes the physical sciences. Biochemistry, and in particular the subdiscipline of proteins, is now being applied to the study of names as physical objects. I'd like to spend the next 20 slides giving you some scientific background for the concept. The major types of biomolecules in mammalian cells are water, which comprises 70% of the cell, proteins, which comprise 18% of the cell, lipids or fats, 5% of the cell, small molecules called metabolites and ions, 4% of the cell, carbohydrates or sugars, 2% of the cell, and nucleic acids such as DNA and RNA, 1% of the cell. Now, proteins, which is the major biomolecule second only to water in the cell, are polymers of monomeric units called amino acids. They are analogous to a bead necklace where the beads represent amino acids. So the picture at the bottom of the slide, these spherical turquoise-colored beads would represent amino acids. The points of contact between the beads represent chemical bonds that link successive amino acids in the polymer. And by convention, these amino acid polymers have two names. If they contain 50 or less amino acids, they are called peptides, or if they contain more than 50 amino acids, they are called proteins. However, this definition is flexible. For example, the human hormone insulin, commonly associated with the disease diabetes, contains 51 amino acids. So this places it at the borderline of being called a peptide or a protein. Now, eukaryotes or higher organisms have proteins that have an average size of 472 amino acids, whereas proteins of smaller organisms have average sizes of 283 to 320 amino acids. Within naturally occurring proteins, it has been found that there are 22 different types, and by that I mean chemical structures, of amino acids, and they can occur in any possible combination. The bead necklace analogy suggested that amino acids are all the same size and shape, but in reality, the 22 amino acids found in naturally occurring proteins are very different from each other in terms of size, shape, chemical, and physical properties. The International Union of Pure and Applied Chemistry, abbreviated IUPAC, is the organization that officially assigns names to all chemical entities. And this makes it easier for researchers throughout the world to communicate with each other. Each of the 22 amino acids has been assigned a long and usually complicated systematic name. For example, one of the 22 amino acids is shown as a two-dimensional structure, chemical structure at the top of this slide. The IUPAC systematic name for that is 2-amino 3,1-H imidazole 4-ioyl propanoic acid, a very long and complicated name. Biochemists often use trivial names instead of these systematic names when describing amino acids because they're shorter and easier to use. The trivial name for this same structure is histidine. When describing long sequences of amino acids in a peptide or a protein, these systematic and trivial names can be cumbersome. Consequently, the IUPAC has also assigned three-letter symbols to represent the names of amino acids. So the same structure that I have been showing you has a three-letter symbol, capital HIS, the first three letters of the trivial name histidine. However, even these three-letter symbols can be cumbersome when describing large proteins. The solution for this problem is that since there are 22 types of amino acids that occur in proteins, the IUPAC chose letters of the 26-letter basic Latin alphabet to use as one-letter symbols for the names of amino acids. For example, this same structure that I have been showing you, the one-letter IUPAC symbol for that structure is capital letter H. And this slide lists all of the 22 amino acids that are found in naturally occurring proteins. It lists their trivial names and also the one-letter symbols that represent those trivial names. So in the left-hand column, we have the letters A through M, excluding the letters B and J. And in the second column, the trivial names associated with those one-letter symbols. In the third column from the left, we have the letters N through Y, excluding the letters X and Z. And in the column on the right-hand side, the trivial names associated with those one-letter symbols. What about the remaining four letters of the basic Latin alphabet? The letters B, X, and Z have ambiguous representations. In other words, they can represent more than one type of amino acid. And the letter J has not yet been assigned to any amino acid. The IUPAC one-letter symbols are the method used most commonly in the scientific literature to describe amino acid sequences, and also the method used to store the amino acid sequences of more than 500 million proteins in computer databases. Coming back to onomastics, using the 22 unambiguous IUPAC one-letter symbols, it is now possible to study names from the biochemical perspective as their peptide equivalents. For example, Walmart, WALMART, is the name of the largest retail company in the USA. The letters of the name Walmart, WALMART, could represent the sequence of a 7-amino acid peptide. And this slide lists the one-letter symbols in the name, WALMART, and the associated amino acid trivial names. So W is tryptophan, A is alanine, L is leucine, M is methionine, R is arginine, and T is threonine. Once a sequence of amino acids, in other words, a name, has been chosen or designed, it is possible to subject it to numerous types of theoretical studies. For example, last year I asked the question, does the name or the sequence of amino acids occur within any known protein? And the simplest way to determine that is to do a so-called blast search of protein databases. It was found that peptide Walmart, or the sequence of letters WALMART, occurs in 833 proteins of the National Institutes of Health non-redundant protein database containing more than 500 million proteins. Another type of theoretical study that can be done is to make molecular models of the peptides. And this was done in 2020, and one of the models generated is shown at the bottom of this slide. If we look on the left-hand side of this stick figure diagram for peptide Walmart, we can see the letter W adjacent to the structure of tryptophan. If we move slightly to the right and down, we have the letter A adjacent to the structure representing the amino acid alanine. Continuing to the right and moving up, we have the letter L representing the amino acid leucine. Continuing to the right and down, we have the letter M representing the amino acid methionine. Continuing to the right, again, we have another alanine, the letter A. Continuing further to the right and down, we have the letter R representing the amino acid arginine. And finally, on the right-hand end, we have the letter T representing the amino acid threonine. The size or length of this model is 23 angstroms, or 23 times 10 to the minus 10 meters, or 2.3 nanometers, which places this model and also peptide Walmart in the realm of nanotechnology. Having generated that peptide, we can then do theoretical studies with it. And one question was asked in 2020 was, would it be possible for peptide Walmart to prevent infection with the severe acute respiratory syndrome coronavirus 2, abbreviated SARS-CoV-2 virus, which is thought to be the cause of COVID? And this slide shows on the left-hand side a model of the SARS-CoV-2 virus. We have a reddish-brown sphere in the center, and on the surface of that sphere, there are bluish-green or turquoise-colored projections, and these are called spike proteins. And it is thought that an infection with the SARS-CoV-2 virus, the very first step involves the interaction of those spike proteins with a protein that is on the surfaces of human cells. And what we see on the middle on the right-hand side of this slide are the actual three-dimensional structures determined by X-ray crystallography of a portion of the spike protein, that's the red ribbon, and a portion of the angiotensin converting enzyme 2, that's a blue ribbon, which is the the protein found on the surfaces of human cells. And this model shows the two of these proteins that are bound to each other. If we look at the interface between the two proteins, the so-called interaction surface in this figure, there are certain amino acids that are located both on the spike protein and on the angiotensin enzyme 2, the ACE2 protein on human cells, which must interact with each other in order for the infection process to occur. So the question that was asked is, peptide Walmart capable of binding to those crucial amino acids on the spike protein, thereby preventing it from interacting with the ACE2 protein on the surfaces of human cells and potentially preventing infection of those human cells by the virus? Well, the study that involved a so-called docking program was run on the supercomputer at Boston University, and it was found that peptide Walmart, the model of peptide Walmart, did bind to crucial amino acid residues on the SARS-CoV-2 virus spike protein. And therefore, it was concluded that the peptide Walmart might be capable of preventing infection by the SARS-CoV-2 virus. Other kinds of studies that can be done are experimental, and in 2015, peptide Walmart was synthesized chemically and then tested in university laboratories. It was in vitro studies, it was found to inhibit the proliferation of human female colorectal adenocarcinoma cells and the growth of human male and female urinary bladder carcinoma cells. And also a preliminary test of this synthetic peptide Walmart indicated that it might also inhibit the growth of the bacterium Staphylococcus simulans, which is a known pathogen of skin. Now, other names in terms of their peptide equivalents have been tested and shown similar positive results. And this has occurred in both laboratories that I have been associated with and also in a European lab. So the letter sequences of names now represent a new universe of novel, potentially bioactive peptides. So the concept that I've been describing to you is summarized in this slide. We start with a name, which is simply a sequence of letters, and we look at that name through the lens of the IUPAC unambiguous one-letter symbols for amino acids, and we can interpret that name as a sequence of amino acids, in other words, a peptide. And then that peptide can be subjected to theoretical analysis and or chemical or biological synthesis and testing. So I'd like to thank you for your attention and just mention that references and reprints are available on the website of the Wade Research Foundation and also through email requests to me at the email address of the Wade Research Foundation or through my email address at New Mexico State University. Expensive. And of course, you have to have laboratories where you can test it. So since I don't have my own laboratory, occasionally I do have enough funds to make the peptide or get it synthesized and then collaborate with people who actually do the testing. So, I can't make them as frequently as I would like. If I had the resources of someone like Elon Musk, though, I could, make plenty of them.
Well, we'll see what we can send your way. We have a backstage question that says this. So am I understanding correctly that if a name, in quotation marks, if a name string successfully prevents some illness, etc., it's totally coincidental that that name did that? But as your presentation said, it's just a way to come up with strings one might not have thought of and then test them out. So is that, are we following?
Yeah, I think so. I think the assumption there is correct that it is coincidental, although it can, because these amino acids and the monomeric units which form these polymers called peptides and proteins, they are in biological systems. And I can almost guarantee that almost every single peptide that is made will exhibit some form of biological activity. It's just they're very, very they're part of the biological system. So it is coincidental, but I could say that every peptide will exhibit some form of biological activity. It just may take you a while to find that activity. But I forgot the second part of the question, sorry.
Well, it was saying this, that if one of these strings comes up to be something that prevents an illness, it's just kind of a coincidence, because we were just using that as the name for something we were testing.
Yes, I think, yes, it is a coincidence. Yeah.
Thanks. We have a question from Laurel, and I know you're here, Laurel, if you want to pose that by speaking it.
There we go. Sorry, I had to unmute myself. Hi, David.
Hi.
So my line of work is in naming and branding. So that's what I deal with all the time. So is trademark ever an issue? And what does Walmart think about using their name like this?
Well, after going through all the process of creating it and testing it and getting the publication ready, I notified Walmart about that and asked if they would be willing to finance further research. And they said that's not a part of their job, their job goals. I contacted someone in the head of the organization.
Mm-hmm.
I don't remember who, and they accepted the fact. They didn't say anything about. They didn't tell me to stop that kind of work. They just said that they could not finance it. They weren't interested in financing something like that.
Oh, that's great for asking. Like, that's terrific. And what a missed opportunity for them. It's not like they don't have the money to do it. Geez. Yeah. It's interesting that they don't object to it. I mean, not that there's anything objectionable, but companies tend to be pretty protective of their trademarks and, historically have slapped people down for using them for things. So, good for them for not telling you, no, you can't use it.
Yeah, I haven't published any results that would indicate that there's anything detrimental to the peptide. So maybe that's the reason why.
Yeah, good. That's great. Thank you very much.
You're welcome. Thank you.
David, maybe try naming the next one Apple and see if you get any better results.
Yeah, I did send a proposal to the head of Apple, whose name I forget right now, and I never got a response. I'm working on Walgreens now.
Well, seriously, you would think that perhaps some of the pharmaceutical industry might be interested in something, but I suppose it would matter too. The question that we had, is it just a coincidence if this one works? They might be a lot happier if they turn out to be the one that is the one that works.
Yes, I keep that in mind.
Yeah. See what you can predict.
Yes.
Well, before we take a short break, I just want to comment, Louisa, you gave us the best possible combination for closing the conference with this panel. This is the only panel in the entire 3 days that has been able to combine these 4 topics: poetry, film, sports, and biochemistry. So you wrapped it all up in a delightful way to end. Oh, wait, before we quite close, let me think. We have
Laura. It's Laura, no? We have to thank Laura for that.
We have to thank Laura. It was Laura who put these 3 together. These 4 together. What a delight it has been to close it out this way. And Hoyo Kunmi has given us a comment. Timothy Donald Cook, born December 1st of 1960, is an American business executive who has been the chief executive officer of Apple Incorporated. So maybe that's going to be your next request.
Yeah.